Hello, my name is Jack Boulay and I'd like to share with you my ketamine research. Ketamine has been used anesthetically since 1970. It was a very important battlefield drug in the Vietnam War. Since 2000, its antidepressant properties have been researched, and in 2019, the first ketamine-based antidepressant was approved by the FDA. Relative to other drugs, it's safe, inexpensive, and fast-acting. The mechanism of action of ketamine is largely unknown. What is known is that it binds to NMDA receptors inside and outside the cell, and this is believed to trigger the psychoactive effects of ketamine. The goal of this research project was to understand how ketamine works to trigger psychoactive effects. Now, as stated earlier, ketamine is safe, inexpensive, common, and fast-acting, so it's an especially important drug to research and understand. In this experiment, we used fluorescent imaging to track ketamine within the cell so that we could better understand its localization within the cell. And this is a very new method of analysis for ketamine. With the information we obtained, we have helped expand the scope of ketamine research such that future research may use our discoveries as a basis for their hypotheses and build upon them. And hopefully and plausibly, our research will help improve ketamine and non-ketamine drugs by determining what makes ketamine so superior and capitalizing on those aspects. So I told you that we tracked ketamine via fluorescent imaging. The reason that this hasn't been done before is because in order to do this, a fluorophore has to be attached to ketamine, and this isn't possible without some modifications to the structure of ketamine. In a cell, ketamine metabolizes very quickly to produce norketamine, which greatly shares in ketamine's psychoactivity. And what we did was form norketamine from ketamine, and then we formed two potential analogs, ANK and ANK amide, from norketamine, and we attached a fluorophore to these two analogs, and we tracked these inside the cell. Now, in order to verify that these two possible analogs are, in fact, analogous to ketamine, we tested psychoactivity and voltage dependence. First, for psychoactivity, we placed control mice into a new environment and gauged their locomotor activity and rearing behavior. That is to say, their tendency to explore and to stand upright in a new environment. And we then injected mice with ketamine, norketamine, and the two analogs, and we gauged their locomotor activity and rearing behavior. Our findings indicate that all of the drugs except for ANK amide are viable antidepressants because the mice acted similarly, increased locomotor action and decreased rearing behavior. They were less nervous in this new environment. Next, we conducted a forced swim test, abbreviated FST. In an FST, mice are placed into an inescapable bath for 6 minutes. Those which are given antidepressants take longer to reach immobility time, which is when they stop moving beyond that which is necessary to keep their head afloat, as shown on the right. Upon giving the mice ANK, they were slower to reach immobility time. They were more determined to escape. ANK amide was relatively ineffective. We then tested the voltage dependence of ANK and norketamine. At -70 millivolts, ANK and norketamine both displayed identical capacity to attach to the NMDARs, but at +50 millivolts, the percent attachment by ANK substantially exceeded that of norketamine, suggesting that the ability of ANK to attach to NMDARs is less voltage dependent. Overall, however, ANK did seem to be an adequate analog of ketamine, so we proceeded with our research. Next, we compared the intracellular accumulation of ANK and ANK amide as well. We placed neurons into a sodium chloride solution, NaCl, with either ANK or ANK amide, and ANK accumulation exceeded that of ANK amide. We hypothesized that the reason for this was because ANK is slightly positive and the cell membrane is slightly negative, so they attract one another, whereas ANK amide is neutral. Therefore, ANK would be attracted to the cell membrane and ANK amide wouldn't. We then made the membrane potential more neutral, so less negative, by using a potassium chloride solution, KCl, and we expected that ANK accumulation would decrease. However, we observed opposite behavior. The disparity between ANK and ANK amide accumulation actually increased, so this was a counterintuitive correlation. And we also tested previous hypotheses, which we found to be somewhat erroneous. Some scientists believe that ketamine accumulated only or even just primarily around nascent NMDARs located around the endoplasmic reticula of interneurons. Our findings, however, indicated that none of these three propositions are true. We obtained live neurons and we incubated them for 1 hour in ANK or ANK amide, and the fluorescent imaging revealed that ketamine accumulated in all neurons and not only around the endoplasmic reticula. Next, we performed an experiment with a type of cell called HEX cells, which lack intracellular NMDARs. What we did was we transfected nascent NMDARs into some, and into others we inserted mature NMDARs. Now here, the fluorescent imaging revealed that ketamine does not favor either nascent or mature NMDARs. So these three propositions were shot down. Now, our research team has provided the scientific community with new, visually verifiable data regarding the accumulation and localization of ketamine. Further funding would enable us to advance ketamine research beyond what we have already demonstrated here, and that would in turn benefit pharmacology and anesthesia. In the future, we would like to research whether specific intracellular targets play roles in ketamine's psychoactivity. Although ketamine distributes indiscriminately throughout the cell, it's unlikely that every cellular compartment participates in ketamine's psychoactivity. So we would like the opportunity to discover which compartments are necessary for ketamine's psychoactivity and how so. We also expect to understand why ANK accumulation increased as the membrane potential became depolarized. Now, here are my references for this presentation. Thank you for listening and have a nice day.